our work that I was discussing focused really on a mouse model of Huntington's disease. And it goes back to our original work where we showed that increased cognitive stimulation and physical activity delivered with environmental enrichment shows a dramatic delay in onset in this mouse model of Huntington's. And when we first made that discovery, this was the first evidence in any animal model of a genetic disorder that environmental stimulation can be beneficial. And since that time, we've followed this up in a number of different ways. We've shown that exercise alone can be beneficial in this mouse model of Huntington's disease. We've shown that it's beneficial, both exercise, but also the combination of cognitive stimulation, physical activity, beneficial not just slowing down the movement disorder, but also beneficial with respect to the cognitive deficits, modelling dementia within Huntington's disease, but also in the depression-like behaviours in Huntington's. We're the first to show that you could actually model depression in this mouse model, and depression occurs in approximately 50% of those with the Huntington's disease gene. We also showed later that increased stress, chronic stress, or simply increased stress hormone in this mice could also accelerate onset of cognitive deficits, modelling dementia in this mouse model. And then we've gone on to show various molecular cellular mechanisms, but also most recently we've been able to show that the gene mutation, which in patients we have the same gene in the mice, the gene fragment with the gene mutation, it's a genetic stutter, this tandem repeat mutation, and it affects not just the brain, but it affects the body as well because the genes expressed throughout the brain and throughout the body. And we've shown this in a number of ways, but most recently we discovered that the gut microbiome, which is essentially these trillions of bacteria in humans that occupy the gastrointestinal tract, which outnumber the number of cells actually in the human body. We have about 1 trillion cells in our bodies. We have trillions of bacteria within our gut. So if you just simply count the cells, we're more bacterial than we are human. And this has been shown in other disorders like Alzheimer's and Parkinson's, but we're the first to show that in Huntington's disease, the gut microbiota are altered. We've shown this in mice, and now with collaborators in Melbourne, we're just publishing evidence that the gut microbiome is altered in clinical Huntington's as well. And I presented that within my talk. And this is very interesting from the perspective of environmental interactions as well. The idea that these environmental factors like exercise, like stress, but also obviously diet, could affect the brain via the body, for example, via the gut. And we know that these gut microbiota talk to the brain via small molecules released that go through the bloodstream to the brain, but also via the vagal nerve and also via the immune system and other mechanisms. So that's a major area which we've been pursuing as well, which I presented.